All right. Today we're going to talk about FOX04-DRI, DRI. I'm going to start off by saying before we get started, this conversation is educational and research discussion only. We're talking about biology, mechanisms, preclinical research. It is not medical advice. It is not treatment guidance, and it is not an approved therapies. If you're here to understand how the biology works, then you're in the right place. But if you're here looking for shortcuts or promises, this isn't one for you. All right, so now we're going to get into it. Why FOXO4 deserves a real conversation. Today, we are talking about the FOXO4 DRI, and I want to say right up front, FOXO4 dry is one of the most misunderstood peptides in the entire longevity and peptide research space. It is often described as the senescent cell killer, the clean out peptide, a reset button for aging, and none of those descriptions are actually very accurate. What FOXO4 really represents is something more interesting, um, and also a little bit more limited. It's not a miracle. It's not going to be a cure. It's not even a therapy. It's a biological proof of concept. And once you understand that, then we are really getting into what it does. Um, so the senescent versus apoptotic cells. This is the foundation of what it does for you. So before FOXO4 makes any sense, I want to make sure we understand what the two of those are. There's a difference between the senescent cells and the apoptotic cells, and I don't always get that word out right. Sorry, guys. They're not the same thing, and for some people, it gets very confusing, and that's where a lot of the misunderstanding starts. So apoptotic cells are the clean exit. Apoptosis is a programmed cell death. It's the body's clean, controlled way of removing a cell that is damaged, dysfunctional, and no longer needed. An apoptotic cell receives an internal death signal, activates enzymes, um, and it breaks itself down in an orderly way and is quietly just cleared out by the immune system. There's no inflammation, it's not chaotic, there's no collateral damage. It's a very healthy way that the cells clean themselves. It's our body's way of cleaning it. It's how tissue renews, it's how cancer is prevented, it's also the balance, it's how balance is maintained within your body. When apoptosis works properly, the problem cells, they don't linger when they leave. The senescent cells are the stalled exit. The senescent cells are different. They've stopped dividing. It's still metabolically active, and it should undergo apoptosis, but it doesn't. And so instead of dying, it just kind of stays there. So it becomes a zombie cell, for lack of a better way to state it. A lot of people, that's a terminology that a lot of people have used. They're called zombie cells. And over time, it starts signaling, and the cells release inflammatory molecules, enzymes, growth factors, um, what's called the senescence associated uh, secretory phenotype, or SASP. This creates a hostile environment that stresses neighboring healthy cells, disrupts tissue communication, and promotes further dysfunction. Um, so the key distinctions are the apoptic cells are the ones that are dying, and the synthetic cells are alive and shouldn't be. Um, so why the senescence exists at all? And this is the important part. This is the big key here. The senescence is not a mistake. It's a protective stopgap. It's the body saying the cell is damaged. Do not let it replace itself, replicate. That would prevent cancer. The problem happens when the senescent cells start accumulating. Um, the apoptosis stays suppressed and the clearance mechanisms start failing. And that's when the senescence shifts from protective to a pathological. And FOXO4 exists entirely because of that failure to complete the apoptosis. So it's going to help with the zombie cells. Um, and so why some cells refuse to die. So we're going to talk about that. We're going to get into the survival of the into survival biology. The cells have a built-in self-destruct program. Best way to put it. Um, one of the most important regulators is the system in the system is P53. P53 is often called the guardian genome. And when the damage is severe, P53 normally says the cell is done. But in many of the senescent cells, something changes. Another protein, FOX04, binds to PT53. And when FOX04 binds to the P53, P53 gets trapped. Apoptotic signaling is suppressed, and the cell survives when it shouldn't. This FOXO4 P53 interaction becomes a survival lock, not in every cell, but in every senescent cell, but specifically subset that the researchers became extremely interested in it. So FOX04-DRI was created to answer that one question. What happens if we selectively break the FOXO4 P53 interaction? That's what they were after. And that's it. It's not a toxin. It's not chemotherapy. It's not a metabolic disruptor. FOXO4-DRI is a protein interaction disruptor. It uses inverso, so DRI, peptide design, which then increases stability, resists um, enzymatic breakdown, and allows it to persist long enough to act in research models. When FOXO4-DRI binds, P53 is then released. The apoptotic signaling resumes, and in certain synthetic cells, the cells finally get to complete their apoptosis. So it'll finally be allowed to die off. And that's the basic mechanism. So it is the FOXO4, the dry is breaking that P53 link to the FOXO4 so that it can complete its death and not hang out like a zombie. It's not a magic, it's not mystery. That's what its function is supposed to do. It's supposed to break that link. In 2017, studies that changed the conversation, researchers published a study that shook the aging field. So this was very fascinating for me when I was researching. The cell cultures in mouse models, FOXO4-DRI, selectively induced the apoptosis in the senescent cells, largely spared non-senescent cells, improved physical function in aged mice, and reduced chemotherapy-induced damage. So if you already have cancer and you've gone through chemotherapy, I've had several friends that have gone through chemo. I don't know why we all seem to have friends that have cancer. And you come out of it, and you have damaged cells. This is research studies in mice that it actually helps those damaged cells as well. So that was a very big deal. Because up until then, most of the senolytics were blunt instruments. FOXO4-DRI suggested something new. Maybe we don't need to prison the cells. Maybe we just need to remove the survival signal. And that alone was why FOXO4-DRI still matters. That is what its mechanism is. Um, After that paper, FOXO4-DRI appeared in very specific research lanes in reproductive aging models, uh, fibrotic tissue models. Some of these words I'm going to have some problems with, and I'm really sorry. The endothelial and vascular senescence, the fibroblast-driven skin pathology, and deep mechanistic studies on the P53 binding domains. Notice what's missing? There was no rush towards the whole body rejuvenation, systematic anti-aging protocols, or universal senescent cell clearance. Researchers were asking, where does this mechanism actually apply? And that kind of tells you a lot. So where people kind of go wrong, and this is very important, There's no robust clinical trials. It's never been done. But there is no approved dosing, no validated delivery system, no long-term safety data, and no proof of systemic selectivity in humans. So that does matter as you guys are thinking about this. Like most of all the other stuff that we are working on researching to learn about, it's always going to be a lot of the same, but it is something to remember. Not all synthetic cells rely on FOXO4 to survive. Um, Clear one population and the other remains. FOXO4 is not a vacuum cleaner. It's a scalpel, so it's going to work selectively. Um, why FOXO4-DRI still matters. Despite all of that, FOXO4 is incredibly important because it's proved something foundational. The senescent cell survival can be disrupted through precision signaling interference. That insight reshaped the aging research. It inspired better senolytics, small molecule uh, mimetics, and a move away from brute force biology. FOXO4-DRI may never be an actual drug or peptide that is going to be widely used, but it did change the architecture of the field in understanding. Um, so all of this is really important. So I'm going to try to tie this up kind of in a nice, neat little bow. The apoptotic cells are cells that receive the death signals, activate the apoptosis, and are cleanly removed. That's normal and that's very healthy. The synthetic cells are the ones that should undergo the apoptosis but fail to do so, remain alive, and actively disrupt their environment. FOXO4-DRI exists only because of that failure to happen. It doesn't kill cells, it removes the survival lock so that it can finish going through its death cycle. Um, so I think that's a really good way to look at it. I guess a better way a better way to picture it, um, think of it kind of like a fire alarm with an automatic sprinkler. So when the room is unsafe and a system activates, it puts out the fire and it clears the space. The synthetic cells are the rooms where the fire alarm went off, where someone is jammed in the sprinkler valve and the fire doesn't spread, but the room is still filled with smoke. FOXO4 doesn't destroy the building, it removes the jam so the system can finish doing its job. So it aids our body. It doesn't fix it. It gives it the chance to do what it's supposed to do. Um, FOXO4, in my opinion, it's a really neat, elegant science. There is legitimate research on it. It is still very experimental, and I'm really, I think it would be nice. I hate to say it, um, if it was studied a little bit more in human trials to see where that understanding is. As in terms of protocols or anything like that, you would kind of need to search and see what protocols have been done. But bearing in mind that a lot of the protocols that are out there are what somebody believes, and or you might find where it's supposed to be so many kilograms or so many milligrams per kilogram. And a lot of the studies have been done in very small mammals, mice, rats. So if you want to move on to, experimenting in 150-pound pig, pork, small cow, whatever, you need to know its weight, I think, to get the best of it. Most of those things usually you can find something along the lines if you look hard enough. Dr. Seeds might have it in one of his books. I've noticed that he does break it down sometimes into milligrams per kilogram. So know what your kilogram is of the thing that you're researching on, and go from there. I'm not sure if FOXO4 is in this current one, but I know that he's coming out with a new one. So it's important to understand that whatever protocols are out there, they're all very experimental. Everybody is still learning about it. It absolutely has a place to help clear out those cells that are not finishing their death cycle. It is important, um, because things that can slow that down could be stress, it could be chemotherapy, it could be illnesses, um, it just could be the way of life. There's any number of reasons why this doesn't happen. And, if anybody else watches The Walking Dead, we certainly don't want a bunch of zombies inside of us. Um, so I think you just kind of need to think about it that way as you're moving forward with deciding if this is something that you want to bring into your research laboratory or not. Um, so, I think the biggest thing you need to do is ask the questions that you need to ask of yourself. Um, where am I sitting for an age? Because age certainly has a factor in in how well your apoptosis is going on, stressors, illnesses, a lot of that comes into play. So obviously we want our body of our research animal to get through all of that and see how that it goes. Hopefully, um, I've kind of cleared up a little bit. I know it's a little confusing. I tried to make it as into layman terms as I could because the research material is very, very scientific. Um, but I think the biggest takeaways are understanding how it works, that its mechanism is to break a bond so that it can finish, the cells can finish their death cycle and then be replaced with fresh new cells. They replicate. Um, and that's why it's not a systematic thing. It is actively looking for those cells that are not doing their death job. Thanks, guys. We will be back again with other great information, and we'll be talking again soon.